to our video on cell signaling and communication. Today, we're going to talk about specifically PDGF signaling. But before that, I need to cut up some strawberries for my breakfast. Ow! I accidentally cut myself. Well, fortunately, PDGF signaling is all about this. What do I mean by that? Well, right now, my cells are freaking out. Just like that. Well, if I were to explain you what PDGF signaling was, that would be really boring, right? So instead of that, we're going to show you guys a quick analogy of what exactly is going on right now. Okay guys, welcome to our analogy. So this is representing a wound right now. And here we have our cells. Cell number 1 tries pushing the door, closing it, but it doesn't close because the wounds don't heal that fast. Cell number 2 starts helping her, or the cell, and the door still doesn't close. So cell number 3 is now called in, and they all try. And once cell number 4 also helps them, they are able to close the door, so the wound is healed. Okay, cut it.
The PDGF signaling pathway is an example of receptor tyrosine kinase signaling. PDGF, or platelet-derived growth factor, is a hydrophilic ligand, meaning it cannot cross the cell membrane and must bind to an external receptor. The main purpose of the signaling pathway is to stimulate cell proliferation, particularly in fibroblasts and smooth muscle cells, which are crucial for wound healing and blood vessel repair. Receptor tyrosine kinases are a family of receptors that activate intracellular signaling when bound by a ligand. The advantage of RTK signaling is its ability to amplify signals and regulate complex cell processes. However, mutations in RTKS can lead to unregulated cell growth, contributing to diseases like cancer.
Okay everybody, now we're switching from scene 1 to scene 2. When a blood vessel is injured, platelets release PDGF into the surrounding tissue. PDGF diffuses through the extracellular membrane and binds to its specific receptor PDGFR, located on the cell membrane of target cells. By the way, I'm so sorry for the quality of the drawings. I drew this, Isha drew that. I think it's obvious, but anyway. So, once PDGF binds, the receptor undergoes dimerization, meaning 2 receptor proteins come together to form a functional active receptor unit. This activates the tyrosine kinase domain, which phosphorylates itself into a process called autophosphorylation.
And now onto scene 3. Again, I think Zeynep lied to you guys. I drew the first scene, she drew the second both.
That's a lie.
The phosphorylated tyrosine residues serve as a docking site for intracellular signaling proteins. One major pathway activated is the MAPK, or mitogen activated protein kinase cascade, which ultimately leads to changes in gene expression that promote cell division and migration. The final outcome of PDGF signaling is increased cell proliferation and migration. Fibroblasts move toward the site of injury, secrete extracellular matrix proteins, and help rebuild damaged tissue. This is a crucial part of the wound healing process. And now, to prevent excessive cell growth, the PDGF signaling pathway must be regulated. Termination occurs through several mechanisms. Ligand degradation, in which PDGF molecules are broken down or removed from the receptor. Secondly, there is the receptor internalization, where the PDGF receptor is endocytosed and degraded. And thirdly, dephosphorylation, in which enzymes called phosphatases remove phosphate groups, inactivating the signaling cascade. These processes ensure that cells only divide and migrate when necessary, preventing excessive tissue growth or fibrosis. Certain molecules can enhance, act as agonists, or inhibit, act as antagonists for the PDGF pathway. One important antagonist is imatinib, also known as Gleevec. It is a tyrosine kinase inhibitor used to treat cancers like chronic myeloid leukemia. And as we can see here, our antagonist is binding to the receptor, preventing the ligand from coming in. Imatinib works by binding to the tyrosine kinase domain of the PDGF receptor, blocking its ability to phosphorylate and activate intracellular signaling. This prevents uncontrolled cell proliferation, which is a hallmark of certain cancers. Without activation of the MAPK cascade, cancer cells fail to divide and eventually die. This illustrates the importance of cell signaling regulation, not only in normal physiological processes, but also in disease treatment.
Alrighty, to summarize the PDGF signaling pathway, it's a receptor tyrosine kinase pathway that regulates cell proliferation and wound healing. PDGF binds to its receptor, triggering a cascade of intracellular signals that promote cell migration and division. However, unregulated PDGF signaling can lead to excessive cell growth, making it a target for cancer therapies like imatinib. Understanding these pathways is crucial in biology and medicine, as they help us develop treatments for diseases and better comprehend how our cells function. Thank you so much for watching, and we hope you found this explanation of the PDGF signaling pathway helpful.